So hello everyone, my name is Étienne. I'm a PhD student at the University of Sherbrooke, and I'm also the last talk of your day, so bear with me. So today I'll talk to you about identifying the expression determinants of snoRNAs via machine learning. So what are snoRNAs? snoRNAs stands for small nuclear RNAs. These are non-coding RNAs present in all eukaryotes. They're basically separated in two types. So we have the CD box and HECA box snoRNAs, which differ in their structure and function, and also in the motifs or boxes they contain. Their main function is to guide modifications on target RNA to which they bind, for instance, ribosomal RNA. But in recent years, it was also shown that snoRNAs could interact with other targets, such as messenger RNAs and transfer RNAs. And in human, most snoRNAs are encoded within the introns of host genes, and there's also a lot of snoRNAs encoded as intergenic snoRNAs. And I just want to highlight here that snoRNAs exist in multiple copies in the human genome due to duplication events, but they also exist in multiple copies across different eukaryote genomes. So we recently quantified the abundance of snoRNAs in several healthy human tissues via an RNA-seq method called TIGER-seq. And by applying a filter, an abundance filter of greater than 1 transcript per million in at least one of these tissues, we found that only 1/3 of all annotated snoRNA genes were expressed, with obviously remaining 2/3 that were never expressed in human. Another interesting finding we observed is that snoRNAs encoded within the same host, gene did not necessarily correlate in abundance. For instance, a snoRNA encoded within the first intron would be expressed, and then a snoRNA encoded in a further downstream intron would be not expressed, meaning here not detected in our data sets, although they came from the same host gene transcript. So these observations raise multiple questions. The first one being, why do we see such a small proportion of expressed snoRNAs, so only 1/3 of all snoRNA genes? And how can snoRNAs encoded within the same host gene vary so much in abundance? So I'll try to answer these two questions for you today. So what are the known snoRNA expression determinants in the literature right now? So basically, what are the factors regulating the expression status, so whether a snoRNA is expressed or not? It was shown for a small number of CD box snoRNA that their distance to the branch point was critical for their expression. It was also shown for a small number of CD box snoRNA that the formation of a terminal stem was important for their biogenesis. And these two features were then kind of broadly applied to all of the known snoRNAs we know today, although we're not really sure they're reliable to all of these snoRNAs. So our hypothesis is that there are probably many more expression determinants to be discovered using other methods. So the objectives and methodology of my projects are separated in 3 steps. The first one being to collect different input features for all human snoRNAs. These features can either be directly related to the snoRNA, such as the whole structure stability, the terminal stem stability, whether the motifs or boxes are conserved within the snoRNA, and so on. And there's also a lot of extrinsic features, such as the distance of the snoRNA to the branch point or to surrounding exons, what is the host gene function, is the host gene expressed or not, and so on and so forth. The second step is to train different models to predict the expression status of snoRNAs. So basically say, is this snoRNA expressed or not expressed in human tissues based on these different collected features? And if the models perform well, we could then interpret their decisions using SHAP values to give us insight into what are the main expression determinants of snoRNAs. So in the interest of time, I'll skip the first step of this project and I'll go directly to the training of the models. So I trained 5 different model types. And what I did is I tuned their hyperparameters on a 10% tuning set, and I trained the models on 80% training set. And finally, I tested their performance on a remaining 10% test set. But I'm interested not only in predicting the expression status of 10% of snoRNAs, but actually of all 1,541 human snoRNAs. To do so, I did 10 different splits or iterations in which each time a different test set composition was created. And overall, by aggregating all of these test set predictions, I was able to predict the expression status of all snoRNAs once across these iterations. So here I show you the performance of the models in the form of a ROC curve. As you can see, with the high AUC values, we were quite satisfied with all of the models. So we then looked at their average accuracy across the 10 iterations and across the 3 different data sets. And as you can see, 2 models kind of show an increased accuracy on the training set and then a decreased accuracy on the test set, which is a hallmark of overfitting. So we decided to exclude these 2 models and continue with the 3 remaining ones, which were more stable. So we took the logistic regression, separate vector, and random forest for the rest of our analyses. So I'm now at the last part of this project, which was the interpretation of the models. So machine learning models are often represented as black boxes to which we give some kind of input, and then with some kind of magic, they give us our output, which is here whether the snoRNA is expressed or not expressed. Fortunately, in recent years, there's a lot of interpretation methods that have been developed, such as SHAP values or Shapley additive explanations, which help us to shine light on these black boxes and give us insight onto finally on which features do these models rely the most to make their predictions. So SHAP values are computed for each prediction and for each feature of a given prediction, and they can take either negative or positive values. In our case, when they're negative, it means that this feature helps to predict the snoRNA as not expressed. When they're positive, it means that this feature helps to predict the snoRNA as expressed. And when they're around 0, it means that this feature has little or no impact on the prediction. So from these computed SHAP values, I was able to calculate a predictive rank for each feature. And that's what's represented here on the y-axis. So a rank of 1 means that this feature is the most important in a given prediction. And the higher the rank, the least predictive this feature is in a given prediction. On the x-axis, you have all of the different features that could be used by the models. And you'll see appear different predictive rank distributions, such as this one, which contain around 30 dots for the fact that we use 3 models across 10 iterations. And you'll see that the distributions are also ordered by median values. So you can see here on the left that 4 features kind of stand out from the rest. These are the 4 most important expression determinants of snoRNAs. The first one being the box score, which represents or means here that the more conserved the motifs in a snoRNA are, the more likely this snoRNA is to be expressed, whereas conversely, the more degenerate the motifs, the less likely it is to be expressed. It's then followed by the snoRNA whole structure stability and the snoRNA terminal stem stability. Again, here meaning that the more stable the structure and terminal stem, the more likely this snoRNA is to be expressed. And finally, it's followed by whether the host gene is expressed or not. And then you can see that the median keep increasing and increasing, representing the fact that these features are less and less important in the different predictions. And just to highlight here the distance to the branch point, if you remember in my intro, I told you that it's currently assumed to be one of the most important features for regulating the expression of snoRNAs. Well, according to our results, most of our models find this feature really less important than what it's currently reported. Here, I want to show you an example of a host gene, which is Gas5, which encodes 11 snoRNAs with varying expression statuses. So there are 10 snoRNAs, the 10 little orange boxes, which are expressed, and 1 that is not expressed from the same host gene. Interestingly, all of our models are able to accurately predict the expression status of all of these snoRNAs. And that's what's represented here on this decision plot. So basically, each line here represents 1 prediction, and we're actually following the decision process of the predictor. So basically, you can go either towards the left, so towards the not expressed prediction, or towards the right, which is the expressed prediction, and it can be influenced by the different features that are located here. You can see that for the not expressed snoRNA, it's basically predicted as such because it has poorly conserved motifs and also an unstable secondary structure and unstable terminal stem. Whereas conversely, the expressed snoRNAs are all predicted as such because they have conserved motifs and also a stable terminal stem. Finally, I want to show you that we applied our models to other species. First, we validated our models on the mouse species for which we had available tight-seq data sets. Here, what's represented is the actual proportion of expressed and not expressed snoRNAs in the mouse, but our models were actually predicting almost exactly the same proportion, so around 25% of expressed snoRNAs, which is pretty similar to what we see also in humans, around 1/3 here. So based on these results, we decided to apply more broadly our models to other vertebrates, ranging from the chimpanzee to the zebrafish. As you can see, except for the chicken here, most of the vertebrates show a conserved low proportion of expressed snoRNA, so always around 30% of expressed snoRNA. Digging further, you can see that by plotting the proportion of expressed snoRNAs as a function of the number of snoRNAs annotated per species, we see a nice anti-correlation between the two variables, suggesting to us that the more snoRNA duplication events occurred in a given genome, the less likely this genome expresses all of the different snoRNA copies, and which is why we only see 1/3 of snoRNAs that are expressed, and a 2/3 that kind of keep degenerating in their motifs and structure. So with that, I'd like to thank all the members of my lab, the funding sources, and you for your attention. Thank you. Yes?
I think it was really interesting. I was just curious about the annotation quality of the snoRNA genes, things like chicken, like how confident are those annotations?
We're not that confident, to be honest. So yeah, what we think is that most of the annotations, that's why we see that there's like a 2/3 of snoRNA genes that are not expressed. So basically, we're not really sure if these are even snoRNA genes. Sometimes they don't even have the canonical boxes or motifs in the snoRNA, but they're as such in the annotations.
Are those based off of like ensemble?
Yeah, ensemble, yeah.
An ensemble takes every transcript pretty much. There's a lot of stuff.
Yeah, yeah. Thank you. Yes.
It was super interesting. Thank you very much. Are you going to look at those, if there is a sequence signature of degeneration in the non-expressed snoRNA? Like if you see that there are pseudo-snoRNAs, now that you've identified.
Yeah, we looked actually at each of, so I didn't say it exactly in my intro, but in each snoRNA there are like known boxes. So we looked directly at the consensus motifs, like in express and not expressed snoRNAs for these different boxes, and we see like degeneration at almost each position for, let's say, the C box and the D box, but there's no like typical motif degeneration. Thank you.